Alright, so in this video we're going to talk about the vessel contribution to the cardiovascular system, or these series of videos. So we want to look at how the blood vessels change and how these changes assist or are caused by, as well as assist in the movement of the blood through the body. So a nice cartoon here, again, to look at the two circuits, right, and remind you that this is an entirely closed system of tubes, right, that the right side of the heart pushes blood to the lungs, it returns to the left side pushes it out to the body. We want to get it to capillary beds and back, right, as all this is showing. And ultimately, all the blood, no matter where it went out to, is going to come back through vena cava, superior if we're draining the head and arms, and inferior vena cava if we're draining below the diaphragm, and of course also the coronary sinus. So that's a nice simple cartoon to really illustrate this quite well. And again, the point of the whole cardiovascular system is to move blood to the cells so we can get exchange of oxygen and nutrients. So oxygen and nutrients out of the blood, and waste and excesses back to the blood. So I remark here again that we pick up nutrients in the intestines and liver and oxygen in the lungs, and so that then will be routed through the body by the cardiovascular system. We release or dump our carbon dioxide in the lungs and metabolic or chemical waste in the kidneys. So this is kind of fulfilling what we need to accomplish here on this one slide. Okay, so this is just a review for you guys, again, how blood moves to the heart, and that this is a two-pump circuit like you saw in the last cartoon. And so here we have pulmonary, right, is the right side of the heart. Blood returns through the superior vena cava, inferior vena cava, and coronary sinuses. It is low in oxygen and higher in carbon dioxide. It goes through the tricuspid valve, right, and then from into the right ventricle, leaves through the pulmonary semilunar valve or pulmonary valve into the pulmonary artery, right, goes to the lungs, and we get oxygen and give up this carbon dioxide. We return to the left side of the heart, consider the systemic side. Blood from lungs comes through the pulmonary veins to the left atrium, right, passes into the left ventricle through the bicuspid or mitral valve, leaves through the semilunar valve known as the aortic valve, and we're going to get to all of the body, all right, and we've completed our task. So again, just reminding you, you know what blood is, what it carries, what it has in it, what it's capable of doing, what its functions are. You know all about the heart, which moves it. Again, this part of the chapter is devoted to how do we get it to the cells and back to the heart. All right. So leaving the ventricles, I've mentioned this several times, but reinforce here, going away from the ventricles are the arteries, all right? They will leave as singular tubes, but immediately begin branching and branching and branching, because we have to get smaller and smaller in order to get to all parts of the body. So smaller tubes are called arterioles. The arterioles continue getting smaller and smaller and smaller until we get to capillaries, which they are completely and utterly optimized for exchange. And capillaries are so small, as I've mentioned in the blood chapter, that blood cells move through them single file. All right. Now, after the exchange has occurred, right, with oxygen out, nutrients out, waste, and carbon dioxide back in, we have to return to the heart. So going back to the atria, the capillaries start to get larger. They become venules, which get larger and larger and larger, so they finally become veins, and we return through one single tube as we left as one single tube. All right? So names of these tubes are determined by the direction and size. So whether you're an artery or a vein really is a directional term, and whether you're an arterial artery or an arteriole or a vein or a venule or a capillary is the size. All right. So here we're kind of compare and contrast. So here's actual arteries and veins. You have slides for this to look at this next to each other. Here's a cartoon to show this. So there is definitely more pressure in the arteries, right, because they're going away from the ventricles where we've really contracted and had the greatest force or push. So these arteries have to be constructed to withstand this push, all right? We say here that the lumen, right, and this is true for any tube in the body, a lumen is a space in the center of a tube. In this case, it's a space for blood, right? And the lumen will always be smaller in an artery versus an equal vein, okay? And this is why later on we're going to say that more of your blood volume is actually in your venous part of the cardiovascular system, cardiovascular tubes. All right, so every single tube in this cardiovascular system of tubes that's moving blood out to the body and back to the heart has this layer called the interna or intima, and intima, again, intimate, an intimate association with the blood. And the interna is made of specialized epithelial cells known as the endothelial cells, and is called the endothelium. And this again is simple squamous cells with some elastin and collagen, elastin for stretch and collagen for support. So no matter what tube you look at, this innermost lining is going to be the intima, right? Innermost coming in contact. And here we see it in the cartoon. It's generally a very, very thin layer, again, of simple squamous or specialized epithelial cells. The media, which is not in every single vessel, is constructed mostly of smooth muscle, right? So the media is many times thicker in an equal artery and vein. So here's the media in this, and here's the comparable media in the vein, right? So cartoon shows this difference as well. And this smooth muscle is going to be able to, right, contract and make the lumen smaller, or relax and make it larger. So when it contracts and makes it smaller, we call it vasoconstriction. When it relaxes and makes it larger, we call it vasodilation, right? And this, of course, is controlled by the autonomic nervous system. There's a specific area in the brainstem known as a vasomotor control center, and it's assisted by some elastin, which we often find. And elastin gives a kind of crinkly appearance. So this is elastin here, and then there's elastin woven throughout here again, pretty much for the idea, again, of expanding, assisting with the expansion and the relaxation of this layer. All right, the last layer that you could find would be the tunica externa or adventitia in some places, and so the external, the outermost. This is generally connective tissue of elastin and collagen, and shown here in this cartoon, it's this layer in this vessel, and this layer really in this vessel. Okay, and this again with the elastin allows some distension or expansion, and the collagen gives it makes it flexible and allows for some support. All right, so looking at the arteries, and here's the difference. There's elastic arteries, muscular arteries, and here's an arteriole, which is considerably smaller than an artery. All right, but the arteries will lead to the arterioles because I said it's a continuous tube. All right, so overall, right, arteries have to have thick walls to withstand the greater pressure that they will be that blood is flowing with. And elastic or conducting ones are closest to the heart, and they have more elastin, so they have more of this kind of crinkly appearance to them. All right, and they will expand and contract, right? Really, or recoil is a better term because contraction is really something that a muscle does. So expand and recoil with changes in pressure. So when we have systole, they expand. When we have diastole, they recoil. And this helps us with the flow. And I just tell you that as long as a vessel is greater than 10 millimeters in diameter, we consider it an elastic artery, right? So there's a correlation with size. Muscular distributing arteries are further away from the heart because we are getting smaller, as I mentioned here. There's less pressure. And so these arteries are going to need to, right, contract and relax with the tunica media to help control flow, to increase pressure, decrease pressure, as will occur from this contraction and relaxation. And again, we use this term vasoconstrictive and vasodilation. All right, and they have a certain size limitation as well. All right, arterioles generally less than, right, the 30 microns, but you don't have to remember that. They are leading into the capillaries, right? So arterioles, right, still have 3 tunics, so still the 3 layers that we talked about, but they are much thinner. They are very important for slowing blood flow down, and they do this by providing resistance. So as we get smaller and smaller lumens, right, smaller diameters to the space blood is flowing through, the resistance will go up. And this means this will take away some of the force with which the blood is flowing. And the rationale for this is that we need to decrease the pressure here so that we don't rupture a capillary. The arterioles control what capillary beds get perfused, right? And so they are going to have the ability to, again, with the muscle in here, they can contract or relax. And as they contract, they're going to decrease flow into a capillary. If they relax, they will increase flow. And you will have a Physio-X exercise to kind of reinforce that idea, right? As we get closer to the capillaries, only the endothelium or tunica intima is left. And you might have some bands of smooth muscle that work more like sphincters to control flow into the capillaries. All right, so capillaries, which are really where we put a lot of our mileage, because this is the place where we are going to get exchange, and we need to be able to deliver to all, these 50 trillion cells in the body. So again, the term perfusion is referring to the flow that's moving through a capillary. So this allows, again, the movement of blood into these very small tubes, perfusion, right? And then therefore the movement out of these very small tubes of things like oxygen and nutrients. And then when we put exchange, we're remarking that, well, some things leave and some things come back. So we have movement out of the capillaries and into them. And for that reason, that's why we're only going to have one and only one layer, right? Because distance, right, of stuff in here, distance of movement, right, if we made that distance greater by having more and more tunicas or layers to the actual tube, we would have to spend more time trying to let the oxygen move from inside of here to outside of here. So it's really beneficial to have just one layer of cells left in a capillary, right? So they have a greater permeability, in other words, allowing things to move out, and than anything else. And then we also see differences in that permeability depending on where the capillary is. So this is a continuous, fenestrated, and sinusoid. So you'll notice that, yeah, sometimes there's more of a barrier to movement, like the continuous. Sometimes we have holes in the actual membranes of the cells, in the fenestrated, which more things can move. And sometimes there's actually large spaces between the cells, which means that cells themselves can leave the capillaries. So that's why you see sinusoids in the liver, bone marrow, bone, lymph nodes, so that large cells can leave circulation and go out into the tissues. Fenestrated, we're going to see these in the small intestine and kidneys because we have a lot of things that need to move. And then continuous are going to be in places where we have to be a lot more careful about what leaves, right? A lot more control. All right? So speaking of control, here's the nice picture of the arteriole to venule, because that's what's going to happen. And the movement into these capillaries, these very small tubes, is going to occur. So delivery is going to come through an arteriole, and it's going to move then into a capillary, and then the capillary will drain into a venule. All right? But we can actually bypass these capillary beds, and we'll need to from time to time. And we can do that with precapillary sphincters. And we also have these structures called arteriovenous anastomosis, or really a shunt, where we're not sending blood at all into a capillary. So we have a lot of control of blood moving. And of course, here is the real point to that. We need to control this because the tissues have different needs. So again, and you only have so much blood. So we have to be very careful about where blood is going at any given time. So these structures we have here, shown over here, the meta-arteriole and the precapillary sphincters, really act to control or regulate blood flow. And this just discriminates between the two of these. All right? So we can completely bypass or flood a capillary bed depending on our needs.